Thank you. First of all, really, I want to thank you, OHBM, and the organizers, for not only for the kind invitation, but also for the tremendous effort that they put into organizing this wonderful meeting. So it's a real privilege to be here with you today. And so I'm going to start by showing you a picture of a guy. And no, he's not a neuroscientist. And in fact, I expect most of you to recognize him unless you're Italian like me. And this is because in Italy, we all study this man in high school. He is part of our cultural DNA. And his name is Niccolò Machiavelli. You might have heard of him. Well, he's famous for this book he wrote. It's called The Prince, which is like a ruthless manual for kings and rulers. And there's a sentence there that stayed with me since high school. And the sentence goes like this: brutal things should be done all at once and done early. And so in that same spirit, I thought I would start with a few brutal clarifications, so things that can be really dry, maybe trivial or technical, but I promise they will make the whole journey easier, okay? And so I'm going to start here. I'm going to show you this video. And when I refer to functional connectivity, what I mean here is the kind of signal you are seeing there, okay? So this is just slow, spontaneous fluctuations in fMRI signal recorded while a subject is simply at rest, right? You're familiar with this. And we, as a community, right, we use co-fluctuations, or statistical measure of these co-fluctuations, as an index of interregional communication. And we call that functional connectivity, right? Well, as you watch this video, high open important point becomes apparent, and I'm sure many of you know it already, right? Functional connectivity is a clear misnomer. Because, you know, functional connectivity does not provide a map of connection, but it is fundamentally a measure of synchrony. And of course, you know, I'm aware of the research showing that, you know, this is strongly shaped and constrained by the connecton. I'm not disputing that. But keep in mind, and this is really important for what comes next, that fundamentally, functional connectivity is a measure of synchrony between these co-fluctuations, okay? The second point I wanted to clarify is, you know, what does my title even mean, right? It's kind of critical. So I want to explain disconnectivity in the context of my talk does not mean disconnection, but it means dysfunctional connectivity. So changes in connectivity that can be higher or lower, we reference to a condition like, you know, control people or maybe a baseline, right? And the second thing I wanted to, of course, to emphasize, the parentheses you see here are, of course, deliberate. It's not a typo. And they're meant to signal that the mechanisms I'll be discussing today, they're not really limited to pathology. So if you're not into pathology, you're interested in healthy brain function, please stay. Because the biology we are after, the physiology mostly, is important also for healthy brain function, so it goes beyond pathology, okay? Especially in the second part of my talk. All right, so with these brutal clarifications out of the way, let me just now start my story, this journey, okay? Which was really, you know, prompted by a persistent problem we have in the field. We have these ever-increasing powerful tools to map brain function. You know, we have a prime example of this conference, but we don't really know what the signals we map mean, right? And so what we thought like 15 years ago or so is that one way to bridge this explanatory gap here was by implementing whole brain functional measures in model organisms, such as the mouse, where we can access the biology. And, you know, many people have been doing it ever since, of course. We weren't really the first to think of that. But, I mean, if you come to think of it, it is important in two ways, or maybe more, but especially two. First of all, by combining fMRI with multimodal techniques, we can really bridge events at different levels of inquiry, right? And that's very important. And the second, perhaps even more important point, if you do that, is that we can begin, through manipulations, to introduce causality in brain imaging. And causality is very important if you want to go from maps to mechanisms. I hope you all agree with me, okay? And so that's what we set out to do. And so in order to make, given that we wanted this research to have an impact for you know, human brain imaging, right, for the species that matter, us, we implemented, well, the choice we made was really resting state fMRI, right? And that's because it's very practical, especially in humans, and there are these big databases we can refer to, but also because it is, if you know, I think we are seeing this also in this conference still, it is a very mysterious phenomenon. We still do not know what it reflects, what the rhythms that support it are, and so on and so forth. And that to us really was, you know, made it really the perfect phenomenon to study. And despite, and of course, yes, this is to indicate that, you know, we want to go through the mouse to bridge that. And so, despite the, you know, inherent simplicity, conceptual simplicity of mapping resting activity in the mouse, from a technical standpoint, it turned out to be difficult. It took us quite a few years to optimize that. And what you're seeing here is the first example published by MedLab when we were able really to you know, describe the distributed networks in rodents, in the mouse specifically. And what is really fascinating, and I still find it fascinating this day, is that if you look at organization of these networks, both in this study here and then in a number of follow-ups that my lab published, is that basically what emerges is that the rodent brain recapitulates a number of key organizational principles that are present in higher species, especially including humans. And for example, you can see these are like networks that are homotopic, they recapitulate known system of the mouse brain, but we also demonstrated that mice have like a default mode network, they have a salience network, so you can really establish these correspondences. And these correspondences really go beyond networks, but they can also be more subtle, and this is something we published later on. For example, we described that functional connectivity gradients are evolutionarily conserved across species. There's like a major gradient here, which is polymodal, unimodal, another one in mice, another one that goes you know, through the specificity of modality, through the cortex, and even more recently, we demonstrated that the dynamic organization of these networks is also evolutionarily conserved. So basically, there are a number of very simple parsimonious principles that underline the dynamic structure of these networks that apply across the phylogenetic tree, okay? And so I'm going to skip the details of this stuff here because this is a little bit beyond what I want to tell you tonight, but I think the big point here I wanted to deliver to you is this high degree of conservation is very important. And this is important in two ways, I think. First, because the biology we study in these model organisms is relevant for humans, and that's something we have to keep in mind. We have to pay attention to that. And second, because these correspondences are more than just, you know, interesting. They actually open the door to translating, with some caveats, findings across species, okay? And this is really the foundation of much I want to show you tonight. All right, so, so we got this tool, right? We can map these networks in the mouse. So what should we use it for, right? What can we make of this? Well, I think I gave it away in my title, right? One of the first questions was, okay, can we use this approach to understand what are the biological roots of the functional connectivity disruption that is almost invariably associated to every brain disorder out there, right? You guys know. And that's what we did, actually, which brings us to the first chapter of my story, so to speak, quote, which is called Molecular Decoding of Functional Dysconnectivity. And the example I want to show you comes from our research in autism. You don't need to be an expert in autism, just focus on the approach, okay? And a good starting point to introduce what we found is really to summarize what the literature says about functional connectivity or functional disconnectivity in autism. I'm going to tell you, it's very easy. So the answer is, it's decreased or increased or both. And it depends on the data set, it depends on how large the cohort is, and sometimes it's also depending also how you pre-process the data, right? And all of this can feel, let's say, very rich or very confusing depending on your mood, right? And it can be confusing because, for example, what we see so far here is that if there is a single signature of dysfunction, of network dysfunction, that defines autism, fMRI has yet to find it, right? Because look at that, it's all over the place. And so clearly, this prompted a large debate in the field, right? Why can we not see autism with fMRI? Maybe it's the threshold, maybe it's the pipeline, maybe it's the cluster, maybe it's all of this, all of the above. And I remember when I was reading this literature, I was really struck by the disproportionate amount of discussion or the energy actually spent on debating the methods and how little importance, how little attention was given to the biology of autism. I am a biology by training. And because what if these differences are real? What if there are different signatures of dysfunction associated with autism? And if you come to think of it, this alternative explanation is actually quite intuitive. Because autism is a spectrum, we all know that, right? And it is associated with a remarkably heterogeneous biological landscape. For example, we know autism is one of the most heritable disorders, and many genes, many high-confidence genes have been associated with autism, but they all map, more than 120 now, but they all map onto different, very heterogeneous pathways, right? And so I think it's plausible or even likely that this biological diversity can reflect into different patterns of dysconnectivity, right? But how can we rigorously prove that? Okay, doing that in humans is difficult. It's difficult, why? Because autism is genetically complex, I just told you. It is shaped by the environment, and in many individuals, we don't even know what the actual genetic cause for autism is. And so what we thought like 10 years ago or so is, okay, if it can be done in humans, why don't we do it in mice? And the idea we have is very simple. Given that autism is biologically heterogeneous, maybe we can use genetics as a key to disentangle this heterogeneity, right, this complexity. So we can use individual mutations as single tiles on a puzzle, so to speak, okay? Each etiology, each mutation, contributing a piece to a larger picture. And the idea here is that if we can collect enough of this, well, and we can scan them, of course, using fMRI, say 20 or more, we can then reconstruct or get an approximation of the complex biological landscape of autism under very tightly controlled conditions. When I say very tightly controlled, I mean in mice. In mice, you can control beautifully genetics because all mice are, genetically homogeneous. You can control environment because, they grow into the same litter. You can control head motion and all the things that plague human imaging, okay? And that's what we did. And of course, out of this, then we have two scenarios, two main outcomes. One is that under the control condition, we can check whether these dysconnectivity maps converge, right, onto like a single signature, or whether they diverge, okay? Considering that the only variable that matters here is biological heterogeneity. And so that's what we did. We got together with our friend Valerio in Geneva, and we put together these 20 mouse models, different mutations. It took us 10 years. And what we find when we put all together is summarized here. So here there's a bit of a color coding, first of all, throughout my talk, so keep that in mind. Blue means reduced connectivity throughout, okay? Memorize this. And so what you see here is from the top is a view of mouse brain, and these blue spots indicate reduced connectivity. So reduced connectivity in the mutant compared to its own leader mates, okay? It's a difference. And as you can see, there's a bit of a convergence here because all these mice here, this mutation produced like blue in this midline region, which is a default mode network. But as we zoom out, we see that there's a bit of a shift. We go for default mode network to more sensory regions, and then we have other mutations for which there is minimal cortical involvement, but there's also some cortical stuff I'm not showing you here. And then that's by far my favorite group. And yes, red means hyperconnectivity, increased connectivity. Memorize that. Remember, red from now on is more connectivity. And this is my favorite. Why? Because if you put all of this together, this explains very well, I think, a couple of things. First of all, under these control conditions, there is no single signature of disruption for connectivity, okay? That's an important point. I think it doesn't get any more control than this. And second, and I think this explains very well also the inconsistencies in humans, right? Because human studies, you are kind of like unknowingly sampling from this diversity. So no wonder sometimes you get red, sometimes you get blue, and a mix of that, right? However, these data also suggest something else, and which is equally important, in fact, even more important. As I told you, these maps have been acquired under tightly controlled conditions. And yet, we see this spectrum, right? You can see from blue to red. So what's changing here? What is changing is biology, okay? And this is indicating that functional connectivity is, in fact, very sensitive to the underlying biology, okay? And with that comes a big opportunity, because maybe, if this is the case, maybe, just maybe, we can use functional connectivity to identify biologically distinct subtypes of autism, right? And the power of this approach is that it allows us to test specifically these hypotheses. How can we do this? Or how did we do this? Well, we use a deliberately simple, even brutal approach. So we split basically the data set in two. One with predominant hypo-connectivity, blue, one with predominant hyper. And then we asked, do these groups, which we defined only based on connectivity, differ in their biology, right? That's the question we want to test. And our assumption is simple, because if connectivity reflects biology, then these two groups with opposite, there are even opposite patterns, right? Blue and red cancel out. They should differ in their underlying biology, because if not, I mean, it's very unlikely we will ever be able to use connectivity to subtype autism, right, to identify or stratify it. So that's what we did, as I said. We generated these two group maps, and then we can check what mechanisms are associated. It's very easy, because for each of these brains, we have a tag, which is their mechanism, okay? And when we look at the biology, this is kind of complicated and technical. I'm not going to take you through this, but basically using a simple bioinformatic approach, we can understand what kind of mechanisms are represented in the blue group and in the red. And as it turns out, what we see is this. Hypoconnectivity, the blue group, is strongly associated with synaptic signaling, synaptic dysfunction. And red is associated with immune system and also transcriptional dysregulation, which is not reported here. But I mean, what is really important here is that you can see there's a clear dissociation between these two mechanisms, which is cool, which is what we were expecting, right? And this suggests that hypo and hyperconnectivity under these very controlled conditions are associated with different biological pathways. And this, by inference, also suggests that supports the hypothesis we started with, right? Heterogeneity in fMRI connectivity, to some extent, of course, can encode for the underlying biology. But, there's a big but, many of you are thinking it, and we were thinking it as well. Mice, this is all in mice, right? And mice do not have autism. I think we all agree with that, right? And so the question is, does any of this translate to humans, well, to autism, the real, say, autism, right? To address this is something we addressed. We can take advantage of something powerful, because fMRI, as I told you before, can be, with some caveat, translated across species. And so what we did, we have these group maps, hypo and hyper, so we know in the mouse brain which regions tend to be hypo-connected or hyper in the two groups, and we use those maps here as, let's call them dysconnectivity priors, to guide the identification of scans in humans that exhibit hypoconnectivity or hyper in the same regions of the mouse. We use them like masks, and we pick out those scans. And then we asked, first of all, are there such scans? Are there such deviations? And if so, what are the mechanisms associated with them? That's what we did, and that's what we found. As a matter of fact, there are quite a few of these scans, and they range from 15 to 30% of the autistic samples we have. And as you can see, these scans can be identified across a number of sites. The differences are very large, and they are also replicable across between a discovery and replication data set, which is nice because it kind of supports the idea that this could indeed be two different subtypes. But what about molecular mechanisms? Well, when we go to molecular mechanism, what we see, and well, first of all, how did we map molecular mechanism? Well, what we can do here is called spatial gene decoding. Many of you are experts, and in fact, we copied that from you. We emulated your approach. You can basically interrogate what genes are spatially enriched using the Allen Brain Institute ProMost Atlas in these like blue regions and red. So you get to lists and then you decode them. And what we found is that the hypoconnectivity maps in humans are associated with, lo and behold, synaptic signaling and hyperconnectivity immune system. And if your memory is labile like mine, if it's like shaky like me with jet lag and stuff, I just reporting here the corresponding mouse data, right? You can see that there's a big correspondence here. And so basically, we were able to kind of like to close the loop, right? So we have now 2 subtypes in humans that with some degree of confidence, in our opinion, can reflect, based only on functional connectivity, can reflect synaptic alterations or immune-related dysfunction. And what I would like to emphasize here is that what really sets this study apart, this is clearly a proof of concept study. I mean, I want to make too big of a story, but what really sets this study apart from other clustering approaches, which also seen here at OHBM, is that all of this is grounded in biology. I hope this is something that you guys could appreciate based on what I've been telling you. We are not just clustering stuff here, arbitrary statistically features. We have a clear biological, quote-unquote, ground truth in the mouse that gives us, confidence that what we found here is, biologically meaningful, okay? And of course, if some of you may think, okay, this is only 30% of autism, yeah, right, sure. But our expectation is that if we scale up the biology in the mouse, we might even be able to scale up the sort of decoding we get in humans. Still, consider this as a proof of concept, and I hope this really describes the power of these, cross-species approaches. Now, the story doesn't end here, though, because next comes a bigger question, and the question is, and of course it's relevant for physiology mostly, is, okay, what does hypo and hyperconnectivity mean? Exactly, right? In terms of physiology, what's that? Can we explain that? And this is something that we wanted to address, and so this takes me to the second and last chapter of my talk, which I entitled Neurophysiological Decoding of Functional Dysconnectivity, where we deep into the physiology of this. And so some of you might be thinking, as we were thinking, that, okay, so let's, for example, this is easy, right? The blue groups of hyperconnectivity, we know the structure and function are tightly related, so if there's lower connectivity, you might have some sort of, white matter alterations, fewer axon, and so on and so forth. But this is something that we rule out early on. We've been examining also structuring these mice without one exception. There's only one mouse line that is acallosal. All the rest don't show any major anatomic alteration, even when you probe with viruses. So something more subtle must be at play here. But what could it be, right? Because many things could be happening. Sorry, this was, I don't know what's happening. Okay, this was to show you that basically mutant controls are pretty much the same, fine. Okay, so what's going on here? Okay, where do we start if it's not really white matter? Well, when we went back to our subtypes, something stood out, and it is this stuff here. I told you, hypoconnectivity is strongly associated with synaptic dysfunction. And synaptic dysfunction in autism mostly mean one thing, which is hyperexcitability. There's even this theory, excitation inhibition theory for autism. Some of you know that. And this is also consistent with the fact that some of the models, the mouse models that ended up into this group, they have evidence of possible hyperexcitability or genes are associated with the control of E2I balance. And so now we have like a working hypothesis, right? This is something we can test in rodents. We said, okay, and our working hypothesis is simple. So hyperexcitability in the cortex mostly, during development, because this is autism, so it is an early developmental disorder, should somehow magically or through undisclosed mechanism lead to hypoconnectivity. And this is, however far-fetched this could be, or maybe even counterintuitive, it's something we like because it's something we can test in mice, and we did. So, how do we test this? Well, what we did, we used we engineered a mouse line that expresses these receptors called chemogenetic receptors. So it's like a little bit like optogenetics, but you use drugs to activate this. And you can use this basically to dial up or down the excitability of neurons, right? And so we express this receptor in the cortex widely, And this is like deliberate. We wanted to mirror what could be happening, say, in autism, right, when you have like one gene that is missing or not functioning. And then we can do a number of, a sanity check in this mouse model. For example, when we give these actuators, this drug that acts as think of like a on-demand hyperexcitability switch. You can switch it on and off, okay? You can see there's more firing, neural firing, so electrophysiology, which is cool. And also you have a shift in the frequencies towards higher frequency activity. And we like this because both of these are established marker of increased excitability. So far, so good. We have now a remote switch that allows us to render the cortex hyperexcitable on demand, okay? We did it only during the first postnatal week, which in humans correspond to third trimester, gestationally and early infancy. And then we measure a bunch of things. And if you're interested in whatever we found, there's a poster still on, maybe after the drinks, if I have still energy, happy to take you through. But however, there are two things that I wanted to emphasize here that are really important for this story. First of all, when we do an EEG, in adult mice, so mice months later, months after this initial manipulation, which was transient, only developmental, this cortex, as probed by EEG, is still hyperexcitable. You can see there's 1/f changes, Hurst index, a little technical. However, the cortex is hyperexcitable. It means that it never really homeostatically recovers there, which is interesting. And the same mice also have hypoconnectivity. Look at the blue. The coveted blue is there. And so this is nice. This is nice because it aligns with our hypothesis. And in fact, it allows us also to, refine it, right? Because these two phenotypes are found in the same mice. So one may think that one causes the other, right? So it is basically that hypoconnectivity is somehow, or connectivity is inversely related to excitability. And this is something we wanted to test more causally, because again, this is correlational. Same animals have these two phenotypes, but this doesn't, correlation doesn't imply causation. So we say, let's go causal. How do we do that? Well, we engineered another set of studies. Again, we use this tool here, chemogenetic. It's exactly the same approach. So you have this like switch. But here there are three differences. First, we do this in the adult brain. So these mice are wild-type mice, and then we want to manipulate a region. So adult brain. Second thing is we manipulate a single region as opposed to the whole cortex. We decided to use the mouse middle prefrontal cortex. And then we use what we call an acute approach, which is basically pre minus post thing. So you can have like a baseline and see what happens later, and that gives us a lot of power. We can really see causally what's happened, right? Because this is really tightly controlled. And what we wanted to do here, again, just as a refresher, we want to test the hypothesis that when we increase excitability, this time acutely, of the cortical region, we have hypoconnectivity, so connectivity reduction, desynchronization. And to do that, we use two different manipulations. Of course, excitability can be increased by exciting pyramidal neurons or reducing inhibition, and we test them both because that's another interesting question. The question is, do these manipulations converge to produce the same thing, right? So that's what we did. And basically, well, first of all, this is electrophysiology, so these are just sanity checks. So we are measuring in the manipulated region, okay, brain activity. This is firing rate, and as you can see, this is control, and then when you give that equator, firing rate goes up in both manipulations, which is, yeah, sanity check, happy. And we also, as another sanity check, we also measure bold response in the manipulated area, right, where we have the switch. And as you can see here, bold response increases, which makes a lot of sense because there are a number of classical studies showing that local bold activity is strongly linked to high frequency activity. So far, so good. What happens to fMRI connectivity? Yeah, as I like to say when I tell this story around, this is really the first time in my whole career that a prediction of mine turns out to be correct, so better late than never. Because despite this increase of bold, we found that both increased excitation and decreased inhibition produce hypoconnectivity. And you may be wondering, oh yeah, but is it the prefrontal cortex? Yes, it is the prefrontal cortex, but this happens also if you do it in the somatosensory cortex. So this seems to be something that holds across the cortex somehow, okay, at least in these two unimodal, polymodal regions, which is cool. It is cool because it suggests that cortical excitability and fMRI connectivity may indeed be inversely related, right? And this is really important because if this is really a general inverse relationship, then a strong prediction applies. The prediction is, I'm sure you'll figure it out, is that if we do the opposite of this, if we silence, if we suppress excitability, we should have more connectivity then, right? If that's a general thing. And that's something that we did test. We call this silencing. And that's something that turned out to be correct. This is, in fact, published. And you see that we have lower firing, connectivity goes up, and this is also in this paper probed both acutely and chronically. And so all together, we got this possible general rule, right? Whereby, excitability is inversely correlated to fMRI connectivity. Last year, I gave a short talk here, and I defined this less is more and more is less, right? So, there's this inverse correlation. All right. The question, however, here is, and I'm sure many of you are wondering now, and that's also what we wonder is, why? How so? Because a lot of this is counterintuitive, right? More firing is eventually more activity. And so how can more activity produce lower connectivity? Well, to make sense of this, we need to go back to my initial brutal clarifications. fMRI connectivity is a measure of synchrony, right? And synchrony in the brain arises from neural rhythms, neural oscillations mostly. And so what we thought is, okay, let's go measure how neural oscillations change when we produce these manipulations, okay? So we repeated the whole thing using electrophysiology. So what we did, we planted an electrode in the manipulated regions and then another electrode in a terminal of this manipulated region. And then we came up with applied a very simple measure of electrophysiological functional connectivity, which we call coherency. Most of you know what it is. It is basically a measure of how in phase these rhythms are. And of course, this is electrophysiology, so this goes across the rhythms. So let me show you how we interpret. This is really a key graph, so pay attention. So this is frequency. This is this, local field potential is what we measure. Coherence modulation, basically, it's electrophysiological functional connectivity, okay? And basically, 0 is no change. So this is like the change produced by the manipulation. And as you can see, you can tell yourself, right? You don't need to be an expert. These two curves are very different. And so that this is very puzzling because both manipulations produce hypoconnectivity, but these profiles are different. So how can we reconcile this? These don't make sense, right? Well, I think we can reconcile this if we make, well, first of all, if you remember an important point, again, in my clarification, fMRI, these low fluctuations are in fact very slow. And so I think it is reasonable to hypothesize that perhaps what really matters when it comes to fMRI is the leftmost part. So the assumption is that fMRI connectivity is disproportionately sensitive to this part here of the spectrum and relatively insensitive to the rest. And if that is the case, then the prediction is that both curves will be below 0, which means lower coupling into this leftmost part. And this is just a log representation that suggests that we may be onto something. This is exactly what's happening. But that's clearly not enough, because if this is a general rule, then another prediction comes, that when we have more connectivity, hyperconnectivity, you remember I showed you when we silence, we have more connectivity, then we should have more electrophysiological coupling here. And that's exactly what we found, which is again encouraging. And that's perhaps a third prediction here, which is still important. If this coupling in these slow rhythms is the backbone of fMRI connectivity, we expect them basically to co-vary with the magnitude of fMRI. In other words, what we expect is that the bigger this electrophysiological coherency change is, and the bigger should be the fMRI response. And that's exactly what we found, but this should be holding only for the very slow part, which is here between 0.1 hertz and 4, or slow again below 1 hertz, and that doesn't apply to other frequencies, okay? And so if I put all of this together, now we have like a general framework or even model is not the right word, but let me use it, to explain connectivity and its alteration. And I'm going to explain it now, very simple. There are two interconnected pillars. The first one is the one I explained, I think, in great details. Cortical excitability inversely modulate fMRI connectivity. And this is true, and this is happening because of the second pillar. The second pillar is that fMRI connectivity is dominated or supported or, if you like, disproportionately sensitive to very slow rhythms, okay? And mind you, I'm not saying this model does not imply that fMRI connectivity is insensitive to faster activity. It can be influenced and modified somehow by faster activity, but this happens only through interactions between the fast activity and this low backbone, right? So basically, faster rhythms can influence fMRI, but they do so primarily via interference or disruption of this very slow backbone, okay? And of course, first of all, this applies to the cortex, I don't know, we don't even know, this is just, clearly a suggestion. And I know it's very simple and even simplistic, okay? But something I like, something I like about this, though, and I cover some implications in the last slide in a few minutes, but something we like about this sort of like simple model is that it comes with a number of easily testable hypotheses. And before wrapping up and discussing some implication, I want to show you one of these tests. And I think the most easily verifiable hypothesis here is that only these low rhythms should support or entrain fMRI connectivity. If you go faster, nothing happens. And that's something we tested. There was a talk this morning by Elizabeth, so go check it out if you miss it. But basically, what we did here, we used optogenetics to test this hypothesis. But instead of using optogenetics like most people do, like using pulses on, off, right, a square thing, we use optogenetics to produce rhythms. However, there's an important caveat here, or technical point. Rhythms in the brain, I'm sure you know, are nested, right? They're rarely like, single rhythms. And so that's what we did. So we designed pulses to generate a fast carrier frequency, for example, 20 hertz, which is, I think, beta. And then we modulate the power of this to produce a slow envelope frequency, which is another rhythm. You see, in this case, very slow one. We deliver them in the prefrontal cortex, and let's see what happens to connectivity. What happens? Now you're experts. Red means increased connectivity. So when we deliver this, we have more connectivity, which honestly doesn't mean much, right? Because it could be 1 million things. It could be maybe there are downstream effects, neuromodulity, whatever. You should be skeptical. However, what's super exciting is that when you look at the ensuing signal, wait, how do you go? Okay. When you look at this ensuing signal, what happens is that it becomes strongly entrained to what? To the envelope of my rhythm. The entrainment is very tight. We can measure with a metric called phase locking. Many of you know it. And if you map the phase locking, you can see really it's super tight, and it happens throughout all the terminals of the prefrontal cortex. And this is really suggesting, it is in line at least to the idea, that fMRI connectivity is highly sensitive to these slow rhythms. But here, we deliver them both, right? They're nested. There's also a faster component. Does this imply that the faster component is not important? Well, the data we have suggests that's the case. The faster is not playing a role. I'm going to show you how. Well, this is, again, I'm just plotting again what I showed you before. It's exactly the same data set. But here, and this is just to show that the carrier frequency, it's 20 hertz, a single line. In order to test the role of the carrier frequency, what we said, because we're kind of lazy, we said too much work, let's go for something shortcut. We randomize the pulse. If the carrier frequency is important, when we scramble it, we should see something different, right? But if you look at the result, they're exactly the same. You have more connectivity, and this connectivity is still tightly phase locked to the envelope of this scrambled signal. And this is really arguing against the role of faster frequency. So the conclusion here to us, or what we suggest, is that faster frequency carrier do not have much of an impact, okay? Last slide, or almost last, that's another one, is this. okay, so far, this suggests that fMRI is relatively insensitive to these fast rhythms, and it is primarily supported by this slow oscillation, very slow. Then the question is, how slow is slow, right? How fast can we go? And so in the next experiment, the last I'm going to show you, we changed the frequency of the envelope. We went for the slightly faster envelope. We went actually very fast, 4 Hertz. In EEG terms, it's still very slow, but 4 Hertz is not bad. And as you can see, this is also to show you, to signal that for all of this, we have electrophysiology clearly. In Elizabeth's talk, it reported, I think. But anyway, these rhythms, they produce very strong entrainment with neurons, okay? So they work very well. We checked that. However, when you do fMRI, 0.1 Hertz is what I show you here. With 4 Hertz, you don't see any change whatsoever, 0, despite this super strong, massive rhythm that we are generating. And this suggests that 4 Hertz is already too fast. If you're wondering where the cutoff is, Elizabeth did other studies. It's around 1 Hertz. And also to signal one thing that here clearly we checked with ultra-fast MRI to make sure that we are not limited by Nyquist frequency sampling, right? So here we went 65 milliseconds with, I don't remember, 10 Hertz or something, okay? And so basically all of this, and I'm going to really wrap up. I have half a minute or maybe 1 minute. It's supporting this simple, once again, perhaps simplistic model that we are, bringing forward. Should be able now to release all this data as a preprint very soon. And as I said, notwithstanding its limitations, right? I think what we like is that we have now a number of testable hypotheses, and testing is kind of easy. And for one, this also allows us to close the loop. Yes, because when we started with autism, now we can attach also this. We can presume, and this is something we're going to test, by the way, through collaborations, is that this hyperconnectivity can be synaptic, and it can indicate, in fact, hyperexcitability. And hyper can indicate hypoexcitability, okay? But the implications can also go beyond autism, and I'm just maybe in next minute or so, I want maybe to bring up a couple of things for discussion now or later, if you want. For example, think of like when you have stroke, and there's a lot of evidence of hyperconnectivity in the ipsilateral area. This hyperconnectivity, I read reviews, people have been saying, oh, maybe it's like rerouting, maybe it's compensating network. Maybe a very simple explanation is that you have neuronal death, less input, so less excitability, so to speak, okay? Less drive. And the same is for dementia or other, like early dementia, when there is often hyperconnectivity in spite of neuronal loss. This is like perhaps a parsimonious way or and physiologically rooted way to explain those changes. And of course, this is for you guys to test clinically, some stuff we are doing ourselves, and let's see whether this will hold, or maybe we'll have to refine. But I hope you agree with me that if this simple model is validated, perhaps refined, because it is admittedly very simple, it could also have broad implications also for, not only for interpreting disease, but also for interpreting connectivity in the healthy brain, right? Across development, aging, and think of neurostimulation. I think a lot of the stuff I've been seeing with TMS and such could make sense within this simple framework, okay? So I'm going to stop here, and of course I cannot stop before thanking all the great people in my lab that have contributed to this research, both past alumni and the people that are just pushing this forward. And also I have to acknowledge the help, friendship, the ideas of a set of collaborators throughout the world that really gave my lab a little bit of a competitive edge, I guess, because of the passion, the help, the selflessness. And and of course I want to thank all the funding agencies, and thank you all for listening.
Thank you so much. Thank you so much, Alessandro, for a wonderful talk. We have many questions. It really started out with an enthusiastic response. Our first comment, of course, was just, even before you'd even had the first slide up, amazing work. So thank you. Thank you. We don't have time for many questions, but one that was highly voted, and which I'll bring up here. To what extent do you think vascularity rather than neural or hemodynamics drive or confound functional connectivity?
I think that's an important point. All I can say is vascular components need to be studied using the same approach, and they clearly can have an important contribution. And I cannot speak much to that because that's not an angle an area we investigated. However, what I can tell you is that in these manipulations, we have a lot of controls for vascular effects. For example, there are controls when we use drugs that produce the same bold increase, but that does not affect connectivity. So we could rule out a major vascular effect in these manipulations, but this does not speak to what could be like primarily vascular events for which I cannot really say much because I don't have empirical data. But that's clearly an important component.
All right, well, thank you so much for a wonderful talk. Unfortunately, we're out of time, but I'm sure there'll be an opportunity for questions throughout the rest of the conference.
Thank you for having me.
As well as online. Thank you for your talk, and just to the, and again, just a reminder, please share your feedback through the app, and join us in the poster hall now for the reception. Thanks.